Welcome back, ladies and gentlemen. So today we're going to talk about the regulation of the cell cycle, or more specifically, what happens when that regulation doesn't go as planned. So when we look back at the cell cycle itself, remember that there really are these three major checkpoints in that cell cycle. You've got one at the end of your G1 phase, one at the end of G2, and then you have that third final checkpoint at the end of metaphase in the actual mitotic division itself. So those checkpoints are put in place for a multitude of reasons. One, we want to ensure that it's actually necessary to make new cells. Is the cell itself getting too big that it's becoming inefficient in moving its materials in and out of the cell across the cell membrane? Also, your cells, especially at the end of G2, are looking to ensure that once it's gone through that synthesis phase and duplicated its genetic information, has it done accurately so that we're not going and dividing this cell that's now carrying mutations, changes in the DNA that may very well become a problem. And same thing in metaphase, we want to ensure that all of those spindle fibers are properly attached to those chromosomes so that when we move into anaphase, those chromosomes actually split evenly when we go to create those new cells. Problem is that there are definitely mutations that occur, definitely cells that get past those checkpoints even when they shouldn't. So what we're looking at here is that part of that control mechanism, part of what decides molecularly, molecule-wise, whether or not those cells are allowed to go through those checkpoints are things like these growth factors. And these are these big protein groups, categories that your cells will slowly build up a level of those growth factors as they're going through the stages of the cell cycle. And that's what stimulates the next phase in the cycle itself. So building up or getting a concentration of a particular type of growth factor will allow them to go through the G1 phase. You'll get a certain level of growth factors that'll build up as they go through G2 so that they actually go into that mitotic phase. And these things rise and lower as different stages and different phases occur. So those are good things. Those are normal things. But there are situations where these cells will either overproduce these growth factors at the wrong times or they'll just ignore whether or not those growth factors exist. And that's where we start talking about cancer cells. Now, your cells have a mechanism in place to recognize when things have gone awry. And so those damaged cells are supposed to go through this thing called apoptosis. So we hit certain checkpoints, those checkpoints recognize, once again, this is all by molecule presence, that there's damage or these cells aren't performing the function properly that they're supposed to. And so they're basically supposed to hit the self-destruct button. Get destroyed, go through apoptosis, what we know is programmed cell death, so that we can replace that damaged cell with an actual healthy one. When your cells no longer recognize that necessity to self-destruct, to go through apoptosis, that's when we start looking at cancer. And by definition, cancer really is a misregulation of the cell cycle. It's uncontrolled cell division. All right, so that this breakdown of these regulatory methods has occurred. So when we look at discussing the hallmarks of cancer itself, there really are two big principles that your cells, as they grow in a tissue, are supposed to exhibit. So when we talk about growing a tissue, everything should be growing in these flat sheets. All right, so single layers thick, and then the layers of cells build on top of that. So in order for cells to properly divide, and we can observe this in tissue cultures, so in basically petri dishes designed to grow tissues in an incubator, tissues should grow one cell next to the other, right? So sort of filling in this space here. If something were to happen like a paper cut, right, and you damage some of these cells, they go through that apoptotic process because they're now damaged, they die off, they get destroyed, then the cells surrounding that damage should recognize that there's now a space, get stimulated to go through cell division, and fill back in the gap. And that's this idea of density-dependent inhibition, right? So inhibition means to stop something. You're not allowed to proceed. So if we're density dependent, right, we're dependent on the number of cells surrounding us. So they should fill in so that everybody's touching each other, but nobody's crowding over top of each other. When we look at sort of cancers, one of the hallmarks of cancers are these tumors, these masses of cells, these balls, these bundles of cells, right? Okay, and so they grow on top of each other. Why? Because somewhere along the way, a cell had a mutation that now allowed it to ignore this density dependent inhibition. It would continue to grow and grow on top of cells around it, as opposed to recognizing that it is now surrounded by a tissue, surrounded by cells, and stop growing. So there's one piece. Cancer cells don't recognize that density dependent inhibition and create these tumors or these masses. The other thing that often is associated with cancers is the ability for cancers to spread to other parts of your body, what we talk about as a metastasized cancer. And what's happening here, when those cells are able to break off of these tissues, those cells are losing their ability to recognize this thing called anchorage dependence. So your cells are supposed to be attached to some sort of flat surface in order to allow it to divide. So when we talk about it in a tissue culture, it has to be attached to a matrix at the bottom of that petri dish. When we talk about it in your body, remember us talking about that extracellular matrix, sort of that Velcro that holds cells, layers of cells together in a tissue. If they're not attached to that, they shouldn't be dividing. And when that anchorage dependence breaks down, that's when these cancer cells are able to break off of these tissues, end up in things like your bloodstream, and now they can go get lodged in other places in your body. So what might have started as breast cancer now starts popping up tumors in your liver or in your brain, because that cancer is now spreading because it's lost not only the density dependent inhibition, which is why the original tumor got created in the first place, but now they've also lost the anchorage dependence and are able to start spreading. So when we look at those tumors, we classify them either as benign or malignant. Benign tumors, although I put the word harmless here, that's probably a little bit of a misnomer. They're harmless in the sense that they've lost the density dependent inhibition, so those masses will form, but they're a little bit easier to treat because they will stay localized. So we can kind of go in, cut them out, so long as we get all of it out, you should be okay. The malignant tumors are the ones that are harder to treat because these are now the ones that not only have lost the density dependent inhibition, and that's why the tumor is forming in the first place, but they've also lost the anchorage dependence, and so now they can metastasize or break off, and those cancer cells can now end up being carried by your bloodstream and end up somewhere else in your body. All right, so when they start talking some of those higher stages of cancers, it's because they're malignant, and that's where we're getting those issues from those secondary tumors. So let's go back to how cancer loses those abilities, because everything is ultimately controlled by your DNA, right? That DNA's job is to then produce proteins. Those proteins are things like those growth factors that then send signals for your cells to do whatever it is they're supposed to do. All right, and so most of the damage that happens to your DNA happens because of something in your environment. All right, so carcinogens, these are cancer-causing agents. These are things like radioactive material, chemicals, things in your environment that we know will create mutations in your DNA. And if those mutations happen to occur in certain genes that are associated with cancers, then we can talk about that chemical or that radioactive element being a carcinogen. If it's not specifically cancer causing, just it causes a mutation. We'll talk about this word mutagen somewhere along the way this year. And that's just a broader category. So any mutation-causing agent is a mutagen. Anything that is specifically associated with cancer is called a carcinogen. And when we look at genes that are associated specifically with cancer, we really get two major categories out of this. And so one of them are these things called proto-oncogenes. And proto-oncogenes are actually a good thing. Their job is to create these growth factors. And so long as they're doing their job appropriately, they will help to be part of that cycling of concentration of those growth factors to get us through the cell cycle correctly. Problem is that if a carcinogen comes into play and creates a mutation that occurs in a proto-oncogene, it will turn into what we call an oncogene. So drop the proto part. And oncogenes are specifically related to this abnormal stimulation of the cell cycle, right? The breakdown in that cell regulation, and that's where we get the cancer. So here's my normal good functional gene. It's doing what it's supposed to do. But if I get a mutation here, any of these could be considered oncogenes, where they're either not breaking down those growth factors like they're supposed to. They're overproducing growth factors above and beyond what they're supposed to. So that's the kind of stuff that can happen to these particular genes. The other gene that are also often associated with cancers are things called tumor suppressor genes. And once again, these are good functional genes. Their job is to suppress these tumors when they show up. Naturally, every one of us is probably sitting here and at some point in our life have come across a substance or even just a random mutation that has caused a cell in your body to start dividing uncontrollably. But you've got things in your body to say, that's not supposed to be there. Let's get rid of it. And so proto-oncogenes are there to help with that. Your tumor suppressor genes are another. And so they're supposed to help prevent the uncontrolled growth. So these also are things that will show up as part of those checkpoints. So they'll recognize that DNA is damaged and try to go back and repair it. Or they function in the control of cell adhesion, right? So go back to that anchorage dependence. The proteins that are produced from these genes are supposed to ensure that those cells are attached to something. They can inhibit the cell cycle, so stop the production of growth factors, so on and so forth. But if I get a mutation in tumor suppressor gene, all of those helpful control mechanisms break down, and so we can also end up leading to cancer this way. So here's the good news. It's not going to take a single mutation to cause a cell to become cancerous. It takes multiple mutations. And it's not even a couple of mutations in an oncogene or a couple of mutations in a tumor suppressor gene. You need multiple mutations in both those oncogenes and those tumor suppressor genes in order to create the cancer. So when we talk about risk levels for cancer, somebody who has a family history, for example, of breast cancer, is at a higher risk because they're already born with a mutation or two in their DNA. And now they need to come in contact with some sort of carcinogen in their environment that then creates the rest of those mutations to cause the cancer. For somebody who doesn't have that family history, they're at a lower risk because they now need to come in contact with enough carcinogens in their lifetime to create all of those mutations. So I'm going to vastly oversimplify this for a second. Let's assume that to cause breast cancer, it takes 5 mutations, right? If you're already born with one of them, now you only need to come across something that causes 4 more in your body. If you're born with 2 mutations because of your strong family history, now you only need to come across something that's going to create 3 mutations, as opposed to somebody who has no family history of that cancer, so theoretically is carrying no mutations at birth for that cancer. Now they've got to go find 5 more. So hopefully that makes sense that you're at a higher risk, not that you're necessarily guaranteed to get the cancer, even when you have a family history, but we'll be a bit more cognizant about screening you and testing and watching for things when we know that you're at a higher risk. So hopefully we can catch it early if it does in fact appear. So this is kind of what I was just saying, right? So it's possible to inherit mutated genes, but once again, it's not necessarily guaranteed that you're going to end up developing the cancer, even if you're at that higher risk. It's just more likely statistically, right? So once again, we're going to kind of monitor you, be a little more aware. So knowing your family history, if you can, is...
Hi, it's Mr. Anderson. Over 1 million people every year develop cancer. There are over 100 different types of cancer, but many people don't really know what is cancer. And the answer to that question is very simple. If we look at a normal cell, on the inside of the cell is the nuclei, and that contains DNA. And the DNA contains genes that control the actions of the cell. And one of those important actions is how a cell makes a copy of itself, how it goes through what we call the cell cycle. And so if you take a normal cell and put it in culture, it will simply make a copy of itself. And if there's room, those cells will make copies of themselves, and they'll keep doing that until they fill up that area. And if one of these cells dies, then an adjacent cell is going to jump into that cell cycle, and it's going to fill that hole. And so that's how you went from a zygote to the trillions of cells that are inside your body. And that's also how you replace cells. And so your skin is constantly losing cells off the surface, but we're creating cells underneath that. And so the cause of all cancers is the same. There's going to be damage to the DNA. And so that DNA is made up of what are called nucleotides. And there are tons of genes in there that are controlling this cell cycle, when a cell should make a copy of itself and when it should stop dividing. And so a mutation is simply damage to that DNA. Now, it could just occur spontaneously. If we all live long enough, we're all going to develop cancer because these mutations are going to start to accrue during our lifetime. But we can also increase the rate of those mutations using the environment. And so, for example, if we smoke cigarettes, that could cause mutations to the DNA. Increases in UV radiation, we're finding that decreases in exercise, increases in certain types of diet can cause mutations, and that can cause problems in the cell cycle. And so let's say we get a mutation in this cell right here. There are going to be genes within that cell that sense something is wrong, and lots of times it'll undergo apoptosis where that cell dies and another cell moves inside. But during our lifetime, we start to accrue more and more mutations. And eventually, enough mutations build up in one of those cells, and it becomes not a normal cell, but a cancerous cell. And this takes a long time for that to occur. And so this is a pretty awful graph. This is the increase in the popularity of cigarettes. And so this is the number of cigarette smoking per person per year. And so as they were doing that, they were increasing the mutations inside their body. And we can look decades later, we see the causation. We can see an increase in lung cancer in men as well. And so as we make those mistakes in our cell, we form these cancerous cells. Now, what does a cancerous cell do? It doesn't matter that there's cells around it. It's simply going to divide and divide and divide over and over and over again. And eventually what you create is a tumor. Now, there are two types of tumor. If those cells are restricted to that one area and stay within a membrane, we call those benign tumors, and that's not cancer. But if there is no restriction to that area, it becomes what's called a malignant tumor. And that's really a definition of what cancer is. It's when we have these uncontrolled cell growth and those cells that show uncontrolled cell growth spreading throughout our body. And so this is a video of some cancerous cells. And so instead of just growing and doing the jobs of the cell, you can see they're kind of misshapen. And all they do is make copies of themselves and copies of themselves over and over and over again. That's where all the energy is going. Now, if that was restricted to one area, it wouldn't be as big a deal, but they can move to different areas of the body. And so if we have a tumor right here, it can start to invade the tissues around it. And so that tumor can not only get larger, but it can move into new areas. And if that tumor gets into the lymphatic system or the circulatory system, it can move around our body and it can settle somewhere else. And so that's the problem with a malignant tumor. And so we name cancers based on the tissues and the organs that they infect. So this would be lung cancer, but we could also have colon cancer or maybe pancreatic cancer or breast cancer. It's basically named on the tissue of where it is. Now, each of those cancers, even though the cause is the same, damage to the DNA, which produces uncontrolled cell growth, the way they manifest the disease is going to be totally different. And so if you look at this x-ray, we can see this is somebody who has lung cancer, and there's going to be a tumor right there. And what we're finding is not only are all these different types of cancer different, but each of those individual tumors are different. And between two people, those tumors are going to be different. And even within the cells of the tumor, they're going to be different. And so what does that mean? Well, even though we might treat one tumor, we're going to find that within that tumor, there are going to be different cells. Each of those have different genetic problems. And so we could treat most of the tumor, remove most of it, but it's going to come back eventually over time. And so what do we have for treatments? And so since cancer is uncontrolled cell growth, we want to find those cells as quickly as we can. And so screening is the most important thing. Once we find that cancer has developed and we find that tumor, the first thing that we want to do is we want to remove that. And so surgery is generally what happens next. We remove that tumor. We also use radiation. In other words, we're not going to be able to remove all of those cells. There are going to be extraneous cells around it. And so we can target this with ionizing radiation and we can kill all those cells. Now this has dangers as well, and so we try to restrict that to that one area, but it can cause damage to the cells around it. And then oftentimes we'll use chemotherapy. Now what's chemotherapy doing? It's going throughout your whole body and it's targeting cells that are actively dividing, cells that are going through this cell cycle. And so that's why it affects, for example, you get a lot of nausea because it's affecting those cells in your digestive tract, or you lose your hair because it's affecting those cells that are constantly making new hair. And so with all of these, we're getting better and better at treating cancer. What's the future hold? Well, we're really just treating all cancers with these three methods the same. And so what we'll see is if we can get into the genetics and we start to understand how each of those cancers are different, then we can start to target them. And so one of the breakthroughs was Gleevec, which is one of these first drugs that's targeting a specific type of leukemia. And it was affecting a specific machinery within that cell. And so again, the more we understand about cancer, we find that they're all the same, but the causes are going to be different. And so what is cancer? It's simply uncontrolled cell growth, and I hope that was helpful.